Our first speaker today is Associate Professor Tracey Dickson. Tracy heads the research group investigating the cellular mechanisms underlying MND at the Menzies Research Institute at the University of Tasmania. The group's research focuses on using a range of novel in vitro primary culture models in conjunction with transgenic models in human post-mortem analysis. Tracy's goal is to identify novel therapeutic targets through identifying the sequence of pathogenic events occurring in MND. Today she is speaking on mice, models and molecules in MND research, looking for new ways to study disease and at the same time making the most of what we have. Please welcome Tracy.
So thanks everybody for coming today, and as Stephanie said, my name's Tracy Dixon. Normally I'd be talking to you about the research that we're performing in our lab down at Menzies, but today I've been given a different task, and Stephanie said, I would like you to do something a little bit different. Could you just talk about the models of MND, their strengths, their weaknesses, why there's been a problem translating the many drugs that have been positive in the laboratory into a cure, just stuff like that. And can you do it in 20 minutes? And so this is going to be a whirlwind attempt at that. Of course, that is one of the perplexing questions that is really playing at all of our minds. How do we get a drug into the clinic? How can we make what we're doing in the lab translate? So when I was given that task, I was like any good student given a task. I went to the literature, and I didn't have a talk on this just sitting in my repertoire, so I did a lot of reading. And I want to acknowledge that some of the really fantastic reviews on this topic that have come out in the last couple of years are by Australian authors, and some of them are probably in the crowd here. And there's been some really great work reflecting on the models that we do have and the tools that we've got available to us, and the new tools that are being developed all the time for tackling MND. So why do we need models? This little cartoon basically says, I go home today, they cured me using this new miracle drug. I'm afraid it's going to be years before it's approved for humans. And it's a little joke, I guess, on one hand, but on the other hand, it's a real dilemma that we have in motor neuron disease. Not only is there a delay in time, but there's a delay full stop. And in order to sort of do a better job at that, we really need to have the most effective models, and we really need to use them in the best way. And they're the sorts of things that I'm going to be talking about today. So why do we even need models? What's this whole thing about models all about? Well, the thing is about the human CNS is that it's difficult to get to. And so we can't just take out pieces of people's brains and do all of our experiments on them. That's impossible. So we need to get around that. And so we need models so that we can identify therapeutic targets. We need models so that we can perform preclinical trials before we use those promising or candidate drugs on people. So this is just a list, you don't need to read it, but it shows you how animal models have really contributed to the many discoveries that have happened, and it lists the medical Nobel Prizes for medicine and physiology since 1904, and those that have been reliant on experiments that have been performed on animals. So there's a critical role for models in our understanding of disease. But like everything in life, we need to make sensible choices. And really, when it comes to animal models, we have a few ways that we can go. So we can have a model that reproduces the symptoms or the pathological features of the disorder, but it may not necessarily be occurring by mechanisms that are occurring in the human. Or alternatively, we may have a model that's known to be based on known genetic mutations or pathophysiological mechanisms, but that model may not in fact reproduce all the features seen in the patients. So that's the dilemma and the series of choices. We also have a range of different models that we can choose from, and there's a number of different models that are used across medical research for a range of different questions. But I guess when we think about motor neuron disease, they really fall into these main categories that are illustrated here. We have a range of different studies that have been done using C. elegans or the roundworm, Drosophila or the fruit fly, the zebrafish models, and also the mouse and rodent models, the rats. And that's what I'm going to focus our talk on today, just reflecting on those models, and also at the end talking about new cellular models and how we may be able to use those in our research. So first of all, these two guys, Many people in the audience who don't come from a research background are thinking, what on earth are they doing? Looking at flies, looking at worms to try and understand a human disease. But in fact, these simple invertebrate organisms can tell us a lot about human pathogenesis. And we know that in C. elegans it's been used very successfully in a mutation sense by expressing the SOD1, FUS or TDP43 models. And when we express those mutant proteins in C. elegans, we do see locomotor defects and impaired neural transmission, as we see in people who have this expression of these proteins. Similarly, for the Drosophila, that model's been used successfully in a range of different genetic approaches again, and we see that with the same sort of approaches, we see reduced locomotion, reduced lifespan, and some muscle degeneration, again showing links that are conservative across these simple organisms right up to the complex pathophysiology of the human with regards to what these proteins are doing. So the nervous system of these animals are well characterised. That's definitely a benefit. It's easy to access their nervous system. There's a really fast generation time for working with these organisms, and they're relatively inexpensive. So these are really great first ports of call when we're asking questions about basic biochemistry, molecular processes, and also signalling. So that's the good news, and this is where people who work on these organisms start to get angry and grumpy, but anyway, I'll just jump in. But there's some downsides of these models, of course. They don't model, of course, the complexity of the mammalian motor system entirely, and we have really limited functional readouts with regards to what's going on with these animals. So what about the zebrafish? We've already heard today about how zebrafish has been used extensively in many of the labs in Australia, and I know there are experts in the audience working with this model, and it's because of its utility, again, for understanding pathogenesis and integrating into these pipelines of discovery through to realisation of function. So again, this is a really valuable model because we know that there's great conservation of the genome of the zebrafish and also the human genome. This model has been used extensively for studies of understanding the development of the central nervous system, and it has this amazing characteristic that many other models don't have in that it's see-through. So you can visualise development, you can visualise different neuronal processes as they're occurring, and the imaging tools are really incredible, the sorts of imaging analysis that you can do. Again, with this animal model, there are motor tests that are really well established, and it really allows for large-scale screening of small molecules. And again, anatomically, it's been proven to be a very good model, particularly of lower motor neuron degeneration. Its variability is really, its effectiveness is really reliant on the approach used for introducing the genetics. And we see that there's quite a variable array of different approaches that can be used, and some of them are much more successful than others. This model doesn't really have the same sort of analogous corticospinal and rubrospinal tracts that you see in the human, and therefore it's not necessarily the perfect model for upper motor neuron disease. And more we're understanding that MND is a complex disorder involving both upper and lower motor neurons. So this graph shows you across all animal research the change or the increase in using rodent versus non-rodent animal models. And you can see in black the really massive increase in using of mice models since the 1990s, and really the plateau of using non-rodent models. And this, again, from a beautiful review that was put out this year by Rachel Tan and her colleagues here in Sydney that looked at mapping in the ALS animal studies. You can again see the same pattern. This huge piece of pie that's dedicated to the mouse studies, and much smaller wedges dedicated to the other models that support that research. And we know that over 4,500 publications have been dedicated to using mouse models of MND, ALS, and you know, there's good reason for that. So what are they? So what's so good about mice? Why are we all beavering away hanging out with mice? What's so good about them? So the good thing about mice is that they have a mammalian system that has really high genetic homology to humans. And so we can use that, we can adapt that, we can play with that to develop models that we hope, and the goal is that will have relevance to human disease. The models that have been developed using mice this way have shown differential capacity, but ability to reproduce motor neuron loss, behavioural deficits, and also pathology to differing extents that really recapitulates what we see in the human condition. So the SOD1 model that we hear about a lot, because it's the one that's been around a lot since 1996, and we heard about the genetic discovery, and very quickly after the gene is discovered, the animal modelling people sweep in and go, right, well, let's make an animal model. And not because it's fun, but because that's the progress that goes with understanding the disease. We find the gene, we then take it to a model system and hope that that progresses our knowledge. So we know that the SOD1 model recapitulates the really progressive ALS phenotype that we see in humans, that we see in people. We see comparable motor loss with ultimately 90% loss of motor neurons in the spinal cord at end stage. But critically in this model, we don't see the cardinal pathological feature, which is the TDP43 aggregate. So all of a sudden, straight away, you can see we have a model that's very good at one part of the pathogenesis, not so good at another part of the pathogenesis. So when TDP43 was discovered, that also brought great promise to the field because people were hoping that we would then get an animal model that solved this sort of this problem. I better get my skates on. So what we found was that some lines, am I okay? Okay. Some lines show motor impairment and mild motor neuron loss and some behavioural deficits. And we see some of the pathology represented in these models with mislocalisation, but cytoplasmic inclusions are generally rare in the range of different TDP43 models, with exception for those that have been engineered to not have the nuclear localisation signal. This is a really, for the people who work on models in the audience, you know, I know that you appreciate that I'm making a range of generalisations to try and cover these many, many models, because of course within the SOD1 there's over 20 different versions of that model within the TDP43, very similar. With C9orf72, again, we were hoping that there would be a real advance with regards to the tools or what was in our toolbox for understanding disease. And what we know with the new models that are coming out all the time with C9orf, that it's a complicated disease, a complicated sort of genetic profile to model, that there is some hope. And we know that when a particular type of way of introducing the C9orf, we see that there is some ALS phenotype recapitulated and some of the TDP43 pathology. And when using a viral vector to introduce the repeats of C9orf, we see that also RNA foci sometimes form and dipeptide inclusions. So this is an advance with regards to being able to mirror some of the pathology. But there are limitations at the moment with regards to our mouse models. Most are reliant on an overexpression model, and they're expensive, they're time intensive. Many of the different models have a pan-neuronal promoter. That means widespread dysfunctional protein is expressed throughout the central nervous system without the sort of limited sort of cell-specific expression that you would see in people. And of course, these are mice that have been genetically engineered to be as similar to each other as possible. But of course, when you look around just our audience, people aren't similar. People are very diverse and we have a very diverse background. And so grappling with the heterogeneity that is the human condition versus the need for a reproducible model is a real challenge. So what also is not so good about mice, and these pie graphs kind of illustrate that really when we're talking about mouse models, we're very much focused on introducing the genetics, the familial causes of the mouse model of ALS into a mouse system. However, you can see in the pie graph that unknown or the sporadic cause of the disease is represented here in the grey, whereas most of the mouse models really focus on those familial causes. And over here you can see, again plotted in blue, is that how much of the mouse work is focused on the SOD model when really the SOD really accounts for such a small slice of the pie. This isn't to say that the SOD model isn't helpful, that the SOD model hasn't provided great insight, because that's not true. I work on the SOD model in my own lab and many of us do. And the SOD model has really revealed some of the intricacies of the pathogenesis of MND. And it's taken us a long way with regards to understanding what is happening with this disease, the non-cell autonomous sort of relationship between the motor neurons and the surrounding cells, and also the hyperexcitability that Steve was talking about this morning. Many of the cardinal features of MND are seen in the SOD model. But I guess I want you to change your thinking a little bit now about, gee, models are problematic too. What about if we're the problem? What about if the people are the problem, or the way they use the model is not that effective? And this is what this really interesting study that was done by ALS TDI in the States and was published in 2014 is getting at the crux of. And listed down here are a whole bunch of drugs that were initially shown to be really promising candidates in a preclinical sense, and they were taken to trial. However, when they were performed with a really strong set of rigorous criteria around how the preclinical trial should be performed, you can see that the effect, their changes in survival, which is plotted down the bottom here, were really minimal with regards to whether or not these were in fact promising drugs. And the only one here, you can see riluzole having a promising effect, but much lower than what was initially published. And many of these other drugs that were shown to be very promising initially preclinically, when they were performed with this new criteria of really trying to standardise how we do preclinical model research, then these drugs weren't as successful. So perhaps the onus is on us a little bit and not just the mice. So in response to that, a range of different guidelines have been developed over the years for us to think a little bit more deeply about how we plan our experiments. And doing animal work is a privilege, and sitting over the top of that is really a large framework of ethical guidelines without even considering the guidelines that sit around working with ALS. And at all times, our work is thinking around these three R's: reduction, replacement, refinement. How can we reduce the number of animals we use? How can we refine the experiments that we're performing in order to maximise the data that comes with them? And how can we replace animal models? But more recently, or I guess over the last 10 to 15 years, people have been thinking about whether or not there's a set of guidelines that we can be using when particularly handling the ALS model mice. And this really sits around particularly using the SOD colonies. So this is just to touch on, I guess, some of the things that people are really thinking that we need greater rigour around in order to try and do a better job at getting our drugs through this pipeline. So instead of focusing our studies just on male mice, and that's how it used to be for a long time, now suggesting, of course, that gender balance is required, because we know that these disease could have different effects depending on the gender. Looking at the litters and making sure we have balance of the litter mates across cohorts. An important one, of course, is tracking the transgene, as we know, particularly with the SOD1, that this transgene can be lost over time in these colonies, so you can have animals expressing a very high copy number of the gene and others very low. And of course, when you're trialling a drug, you could then misinterpret your results because of that. And that was a lesson learnt in my lab very expensively, very early on, when you do a test and you find that your SOD model's really not behaving the way it should, and it's because the transgene or the copy number has slipped. Really trying to get agreement over what the best time to start treatment is. You know, whether or not a pre-symptomatic trial is of any value, because we know that in humans we'd be starting treatment when they're clinically present. But these are animals that are being driven to express a protein at a much higher level, so is it fair to expect them to be the same? And maybe an initial screen at symptom onset and then going back and doing a preclinical trial, a pre-symptomatic trial. And then a number of different things around basically managing colony management much more strictly, and how we apply statistics and the blinding of investigators to different genotypes and treatment groups, so that we can ensure that bias isn't creeping into our analysis and our perceptions of how these drugs are working. So more recently, a new model has come on the scene, and it comes with great excitement, as all new models do, and sort of great fanfare for the field of whether or not we can get rid of the mice. Can we have a cellular approach to discovery that allows us to understand this disease, that allows us to trial drugs effectively? And I guess my sense is probably a little bit of yes, a little bit of no. So the model, of course, as many of you know, involves taking a skin cell from an actual person, reprogramming it and converting it back to its induced pluripotent stem cell status, bringing it back up to a neuron, and then using that for either a high-put drug screening, trialling candidate drugs, or in this schematic, it's suggesting that you can also use it as a patient-specific therapy. So this, of course, would be a shortcut to trial, and it's been trialled for some of the work done in the States coming out of Kevin Egan's lab, using an iPSC model as a way of circumventing the traditional trials in the animal models. So these iPSCs, the great news is that we show that using this approach, we were able to recapitulate a lot of the pathology. So we do see aggregation and mislocalization of TDP43, SOD1 and FUS. And in the C9orf line, or a line of iPSCs or motor neurons derived from a patient who has the C9orf genotype, we see formation of RNA foci. And excitingly, these cells have really functional phenotypes that you would expect to see in a diseased motor neuron as well. So we see increased cell death, neurofilamentous disorganisation, defects in transport, changes in excitability. And in the field that I work in, most excitingly, what's been seen also is that these cells also recapitulate an increased hyperexcitability early in the disease, and over in time, we see a decrease in excitability, and that maps very closely to what we see in the patient. The other great thing is they remove the need to express the transgene, and using the CRISPR-Cas9 technology, there's the capacity to do a correction of the mutation. And so you can have a line that expresses the pathology, do the correction, and see if you can recover the MND phenotype. So it becomes a very contained cellular experimental system. But they're not perfect yet either. So the protocols for generating spinal motor neurons are variable, and they're improving all the time, but at the moment they aren't standardised, and so we need to be cautious about that. And maintaining multiple clones for many patients is logistically and financially very challenging. Banking fibroblasts is the first part of the process, and then the hard work is coming when we need to grow these lines and maintain them in a highly standardised way and use them in the experiments. So these will be challenges that we will need to grapple with over the upcoming years. But great excitement also sits in the field with regards to, can we not only improve where we're getting our cells from, but can we improve the organisation of these cellular models to more closely look like what you would see in the central nervous system? And many people are working on, including in our lab, microfluidic approaches to try and reorganise cells with regards to looking like they would do in our central nervous system, spinal motor neurons in contact with astrocytes, innervating muscles, and also these ideas of developing organoids or multicellular systems that sort of mirror the lamina that you see in the brain, particularly the cortex. I'm right on time. So the challenges and the opportunities. This is a really beautiful summary diagram, I guess, that shows you that it's not simple, and it plotted across the top of the type of models we've talked about today, not covering the yeast. But down here we can see all the different parameters: life cycle, lifespan, protein conservation, ability to screen compounds, costs, and how much they're utilised. Down here maps, an idea about what these models are best suited for, and thinking about throughput versus relevance for humans. And I think this review makes a really beautiful case that all models have a place in the journey along the pipeline of drug discovery to the clinic. And it's just using them sensibly and in collaboration that is going to end up with the best results. So discovery in the lower vertebrate and simpler models, trialling in the rodent models, also checking our results in a humanised cellular model is by far, I think, the best approach for us to really progress our targets. So I wanted to finish with this slide. This is an opportunity for me to introduce gratuitously a new member of our lab in Tasmania. This is Catherine Blizzard, who received the Laidlaw Fellowship last year, and she would normally be here with bells on, as many of you in the audience know. But she has a new baby who is here, Flynn, and here he is having his first bath. But the message is, it's kind of a wacky style of bathing babies now. When I saw this, I was slightly freaked out. But anyway, I'm sure she knows what she's doing. Don't throw the baby out with the bathwater. And it's kind of a joke, but it really is a true message around models of MND. So we have to always be looking for new models. We have to always be reflecting on how we use our models and be critical of ourselves and how we apply them. But also, I don't think we can assume always that the newest model is the best model, and that we should look at them as ways of adding to our toolbox rather than something new in, something old out. Because I think, as Ian said this morning, this is a complex disease. It's very heterogeneous, and the more models that we can have, the more people with the expertise to use them in a really thorough and stringent manner, I really feel confident that together we will be able to progress with our understanding of this disease and also progressing candidates to the clinic. So thanks for your time. I think we're still at the point where people are looking for a reason to say this is working or this is doing what we're expecting. So, you know, you put in a single mutation, then you sometimes are seeing the gamut of changes. But at other times, you can put in the same mutation and you don't see those gamut of changes in the same cell approach. So I don't think it's as simple yet as this is what a SOD1 mutation looks like in an iPSC. I think it's too variable at the moment. I think we're kind of, just as we're getting our head around getting good at doing the mice work, we're going to have to get our skates on pretty quick and get to some consensus around how we're going to grapple with the iPSCs, because I think you're right, if we can come up with good foundation agreement, we can progress much quicker.
I mean, on the point of heterogeneity, you know, everyone in cancer resisted having to deal with heterogeneity for a very long time, and it cost us years, decades even, of understanding.
Yeah.
You really need to embrace the heterogeneity because it's actually a biologically fundamental part of the disease.
Well, I think this is the opportunity that the cells bring us, because with the mice, we have to grapple with that simple system. And so if the cells bring to the table a capacity to look at heterogeneity, well, then that is a real progress. This is something that all neurodegenerative disease researchers are grappling with. It's not just MND, because once these people present, then often those neurons are lost. But it's a huge body of work. People are looking for new imaging approaches, new techniques to look at the earliest symptoms rather than looking at them later. That, I think, is a challenge, but I don't think it should be a challenge that stops us looking for the treatment as well. So knowing when to apply it, that's kind of a, you know, and getting good biomarkers, getting good diagnosis, you know, that remains obviously a massive challenge for the field. I'm not sure I've got the answer.
All right. Thank you, Tracy. Thank you.
Thanks.